Hello everyone. Welcome to the 7th class of Immunology. In this class, we are going to start innate immunity. Innate immunity is a big topic. So we have divided this topic into two video classes. In the first class, we are going to study the barriers, phagocytosis and PRRs. In the next class, we will be discussing about inflammation and some other aspects of innate immunity. So as usual, before starting this class, let us see what we have learned in our previous class. So we discussed cytokines and their properties in the last class. We also discussed the families. We discussed the receptors and signal transduction. We discussed the cytokine antagonists. We discussed the cytokine related diseases and cytokine based therapies. I hope you remember these all topics. If not, then you can just go back and see the video again. Okay, so let's start with the innate immunity. Now, when we see the term innate, the dictionary meaning of innate is that qualities, characteristics, or abilities that are present from birth. So, important part is that these qualities are present from birth rather than acquired or learned through experience. So that means innate immunity is the immunity which is present by birth. Okay, this is present by birth, and this is the ancient form of immunity. This is present in almost all organisms, whether they are even some small microbes or even the vertebrates, lower vertebrates, or plants. It is present everywhere. Now the form may be different. In case of small bacteria, it may be maybe few molecules. It may be few different ways. For example, in case of bacteria, you have the restriction endonucleases. The restriction endonucleases which cleave the viral nucleic acids. Okay, so that is a form of protection, right? If you move slightly higher, then you will see the insects. They have different molecules, antifungal peptides. In plants also, we saw in the first video lecture that there are resins which protect them from the fungal or bacterial infection. In higher vertebrates, we have different form and even more complex form of immunity and innate immunity. So as we move higher on the ladders, we will see that the innate immunity, not only it's becoming complex, it is bringing more and more molecules, it is bringing more and more different type of cells, but also now acquired immunity is also coming up in the picture. All right, but we are here focusing on the innate part of the immunity. Okay, so innate immunity is the immunity which is present by birth. Remember this. Now let us see the key elements of the innate immunity. What are the key elements? Okay, so we have the barriers, and the barriers are the physical barriers. Physical barriers we have the epithelial layers of skin, so that means we have the skin as a barrier, and then we have the mucosa or the glandular tissue. All right, so basically these barriers, they do not allow any foreign particle or pathogen to enter inside our body unless and until there is a breach. Okay, then we have the chemical barriers, and now these chemical barriers are in the form of the acids or antimicrobial proteins and peptides. All right, now we will discuss this. For example, in stomach we have very low pH acid. Okay, it not only helps in digestion, but also whatever microbes they are coming with the food, it cleaves them, right? So this is the chemical barrier. Then we also have the cellular response. In the cellular response, we have different cells. So we have macrophages, right? And then we have the natural killer cells, and then we have the cooperation between the dendritic cells which activate the acquired branch of immunity, that is the T cells and the B cells. So everything we will see in a complete picture, that how one immunity is, or one form of immunity is inducing another form of immunity, and how they are cooperating. Okay, so we will see that, but let us focus on the innate part here. So, if we just do a contrast or comparison between the innate immunity and adaptive immunity, we will find that innate immunity is present from birth. It is present by birth. It is natural or native immunity, and it is the primitive form of immunity. While the adaptive immunity is acquired by exposure to different antigens. So it takes time. As we grow, then our acquired immunity increases, because over the period we get infections or we get vaccines, and through the infection or vaccines our body starts building immunity. So that is adaptive part. But this innate immunity is a natural form of immunity, and it is present by birth. Then immune response is non-specific. That's very important aspect. That means whether it is the bacteria or whether it is the virus, our innate immune response, it's not going to change much. Okay, so it is non-specific. While in case of the adaptive immunity, the immune response is highly specific. So it is tailored. The immune response is tailored as per the kind of antigen which has entered inside our body. All right, then we have quick response within hours or days. So that means not and even within minutes also. So this response is immediate. As soon as our body detects there is some pathogen or there is some foreign entity, then our body starts responding against that. So the response is very quick. While in case of the adaptive immunity, the response is slightly delayed, as it requires time to develop. It's not like within minutes or within hours our acquired immunity starts impacting or attacking the or eliminating the pathogen, but it takes some time, right? Then response duration is short. So it's not like that it will take the innate immunity is activated for long. No, it is the response duration is very short. Immediately the response comes there, while here the response duration is long. And sometimes we have, because of the memory cells, where we have the response and we have the power to get the response maybe through lifelong. All right, then memory of antigen is not formed here in case of the innate immunity, as we just told you. While in case of the adaptive immunity, the memory of the antigen is formed and it is used for subsequent exposure. So next time when the same infection is going to come inside our body, then we have the memory cells, and these memory cells will do or will give the response in a very quick way and in a higher magnitude. Okay, the repeat exposure has the same response. So here the infection is coming again and again and again, and the response is the same. There is no change in response. While here in case of the adaptive immunity, the repeat exposure has heightened and more brutal response. So once it is coming, it's okay, we will treat the infection nicely. But next time if it is coming again, then our body will give very brutal response, and the response will be very heightened as compared to the previous response. Okay, now these are some of the examples of the innate immunity. For example, we have skin as a physical barrier. Then we have the antimicrobial peptides like beta defensins. Then we have the complement systems. We have gastric pH. We have TLRs, phagocytes, NK cells, etc. So now TLRs we will going to discuss in details in today's class only. So TLRs are toll-like receptors. Okay, and these are a kind of PRRs, and we will see them. So these are some of the examples of the innate immunity. While in case of adaptive immunity, we have the T cells and the B cells, and they are the major cells of the adaptive branch. So I hope with this table you have some idea about how innate immunity is different from adaptive immunity, or how adaptive immunity is different from innate immunity. Okay. Now, let us start with the barriers. So, when we see the innate barriers, we have the physical or mechanical barrier, and examples are skin, mucous membranes, or cilia. So basically, they block the entry, they trap the pathogen, and they expel the pathogen. That's how the physical or mechanical barriers work. In case of chemical barriers, we have like sweat, we have gastric acids, we have lysozymes or defensins. So these destroy or inhibit pathogens chemically. So this is the form of the chemical barriers. Then we also have the biological barriers. So biological barriers, the best example is the normal flora of the gut or the skin. So what we have, the friendly microbes which are present in our gut and skin. So they compete with and inhibit pathogens. Okay, and then flushing is another way, a barrier, like in case of tears, saliva, urine, or peristalsis, which wash away or move pathogens out of the body. So that's how the innate barriers are placed in our body. Okay, now we will see one by one. Now, let us start with the physical or the mechanical barrier. So, skin is the first line of defense and it has two distinct layers. If you can see, first is the epidermis, and this epidermis is a thin outer layer of tightly packed epithelial cells, and mostly dead cells are filled with keratin. So our skin has the keratin protein. Now this keratin is the same keratin which your hairs and your nails are also made up of. Okay. Now if you see this diagram, here you will see this is the epidermis. So this part, okay, the outer part, upper part is the epidermis. So this is all epidermis here. All right, and here you can see it's nothing much, but it is just a tightly packed epithelial cells. Okay, then we have the dermis, and dermis is the thicker connective tissue with blood vessels, hair follicles, sebaceous glands, sweat glands, and scattered myeloid leukocytes. For example, DCs. Remember DCs? Dendritic cells. Then we have phages and then mast cells. So here you can see the thicker part. This part is dermis. So you start with here and it goes up to here. The red part, slightly pink part, this is the dermis. This is the dermis part. On in dermis, you have all sort of, the glands and blood vessels and nervous system. Okay, that's how this skin is made up of. Then we have the mucous membrane. Okay, and mucous membrane are the specialized lining. Okay, these are the specialized lining of respiratory, gastrointestinal and urogenital tracts and the ducts of the salivary, lacrimal and mammary glands. So salivary glands, they secrete saliva, lacrimal glands, they secrete tears, and mammary glands, they secrete milk. Okay, so here you can see these are the different mucosa and various types of mucosa. We have nasal mucosa inside the nose, then we have oral mucosa inside our mouth, then we have esophageal mucosa in our esophagus, then we have bronchial mucosa in our respiratory tract, and then we have the gastric mucosa in our stomach. So these are some of the examples. Okay, they are the strong barrier layers of epithelial cells which are held together by tight junctions that prevent pathogens from squeezing between them to enter the body. So what happens here in case of mucosa, suppose this is one cell, okay, and here also we have another cell. Now the boundaries between these two cells, suppose this is cell number 1 and cell number 2, we have the tight junctions, and these tight junctions, they are so tight that any pathogen, if it wants to come and squeeze out from, or wants to just break this barrier or come through this side, it will not come because it is so tight. All right, so I'm sure these tight junctions you must have studied in molecular biology, where in extracellular matrix part, these junctions are described here. Okay, so you can go back there and you can see how tight junctions work like, right? Okay, so we have the skin and we have the mucous membrane, right? This is the physical and the mechanical barrier of our body. These generate active chemicals. Now, important part is these barriers generate active chemicals and biochemical defenses through antimicrobial molecules. So, they not only are the physical barriers, but also they are generating the active chemicals and biochemical defenses through antimicrobial molecules. So, antimicrobial molecules and chemicals are also generated by these barriers, which help to eliminate any infection or pathogen. Okay. Now, let us see the chemical barriers. So, we have a long list of chemical barriers, right? We have lysozyme. So, this lysozyme is present in sweat, tears, saliva, and it breaks down the bacterial wall. Okay. Now, it is effective against bacteria. Then we have defensins. Defensins are produced in skin, mucosa, and semen, and they disrupt the membrane and they have the antimicrobial activity. We have fatty acids produced by the skin glands, which create an acidic environment and they are responsible for the acid mantle on the skin, right? So our skin has a kind of slightly lower pH towards acidity, which creates an acid mantle and which resists the growth of the microbes. Then we have the gastric acid. Now, this is a classic example in stomach. It kills many pathogens and it also activates our gastric enzymes. Then we have the proteases, which are also produced in stomach. It cleaves pathogenic proteins also, and it requires low pH, and this low pH is given by this gastric acid. Then we have organic acids, which are present in sweat and vagina, and the lower pH to inhibit growth, and so the vaginal secretions are usually acidic. Then we have the zinc, and it is present in semen, and it is toxic to many pathogens, and it is again antimicrobial. In mucus, the mucus is secreted by mucosa. It contains antimicrobials and it is a physical barrier too. Then we have CRP, right? And then it is present in blood plasma and it binds to bacterial polysaccharides. It's a kind of opsonin. What is opsonin? We will see that. And it early induced protein in the innate immunity. The CRP is the early induced protein. Then we have the mannose binding lactins. We call it MBL. Mannose binding lactin. Again, it is in plasma and it binds the bacterial polysaccharide and activates complement pathway, right? And it helps in phagocytosis and complement activation. So the CRP and mannose binding lactins, they are present in blood plasma. Then we have the lactoferrin, and this lactoferrin is present in milk and mucosa, and it binds and sequesters iron, limiting growth of bacteria and fungi, and is also disrupts the membrane. Okay. Then we have secretory leukocytes protease inhibitor, which is present in the skin or the mucosa and mucosal glands, and it blocks epithelial infection by bacteria, fungi, viruses, and it is antimicrobial in nature. Then we have S100 proteins like psoriasin and calprotectin. They are present on the skin, on the mucosal or glandular secretions, and they disrupt membranes. They bind and sequesters divalent cations, for example, manganese and zinc, and they limit the growth. And then we have some surfactant proteins A and surfactant protein D. They are present in mucosal secretions, they block bacterial surface components, and they promote phagocytosis. Then we have RAC3 proteins, which are present in intestinal epithelia, and these bind cell wall carbohydrates. They prevent bacterial binding to epithelial cells and they form membrane pores. Then we have cathelicidin, which is LL37, present in the mucosal epithelia. It disrupts membranes and provides intracellular toxicity. Then we have histatins, which are present in saliva, and these are the lactins which binding to the fungal cell wall. And we have dermicidin, which is present in the sweat gland, and it is antibacterial and antifungal. So these are, the list of the known innate chemical barriers, and this list is increasing, every year or every 2 years or every 5 years. We get some new chemicals, or we just see that the particular protein is also working as an innate chemical barrier. So this list, we can see this is, it's going to elaborate, but not very soon, but yes, definitely this list is updated regularly by our researchers. So chemicals, they destroy pathogens directly or marking them for the immune clearance. So we have two ways. Either they will be killed by these chemicals, or the other way is that these chemicals, they bind those pathogens and they mark for their elimination, so that appropriate immune response can be elicited, and then the pathogen can be killed. Now, there are certain examples of the evasion of epithelial barrier defenses. So, we have the barrier defenses, but still there are some smart pathogens which can breach them. For example, influenza virus. This influenza virus, it attaches to the respiratory mucosa, right? Therefore, it's not easily washed away. And you can see that in flu season, a lot of flu is there in our community. Why? Because of this influenza virus. Okay. And then another example is the Neisseria gonorrhoeae. This binds to mucosal epithelial cells of the UG tract. So basically, urogenital tract, UG tract, it binds there, and then it resists washing away. So this is their smart ways of holding inside the body and giving the infection. So there are so many other examples also, but these are just two of them. Then we have the biological barriers, third kind of barrier. We saw the mechanical barrier, we saw the chemical barrier, now it is the biological barrier. And these biological barriers are the living components that help protect against pathogenic invasion. So here you can see these barriers are primarily consist of beneficial microorganisms. So our body has a lot of beneficial microorganisms, and these microorganisms, they inhabit various body surfaces and internal tracts. Okay. Now, the normal flora is also called microbiota. This microbiota, we have trillions of harmless bacteria. Okay. They naturally reside on the skin, urinary, reproductive, and gastrointestinal tracts. Okay. So there are trillions of harmless bacteria, right? Now, they compete with potential pathogens for nutrients and surface space. So here you can see this is the normal flora, and here this is the pathogenic flora. So pathogenic flora is coming and try to get the surface here, but since the surface is already occupied by the normal flora, then it's not getting chance, and that's how we are protected. But in case if our normal flora is reduced, then the pathogenic flora will get chance, and definitely it is going to produce the disease. So this is basically a competition, the competition between the normal flora and the pathogenic flora. Okay. It prevents pathogenic bacteria from gaining a foothold by occupying ecological niches. And some also produce antimicrobial substance. So they are not only helping to, to occupy this space, but they are also producing some antimicrobial substances which are killing this pathogenic flora. So normal flora helps train and modulate the immune system. It promotes tolerance to harmless microbes and readiness against invaders. So these are the three important functions of microbiota inside our body. They help train and modulate the immune system, because our immune system knows that this is harmless. Okay, so it is not going to give an immune response, rather tolerance comes there. And it promotes tolerance to harmless microbes and readiness against invaders. So in case if there is some invaders, then our immune system can easily detect and then start responding. So that is the ability of our immune system, which can recognize between the harmless and the harmful bacteria. All right. Okay. Now, the fourth kind of barrier is innate flushing. So we have the cilia and we have the mucous movement. So here you see many mucous membranes have cilia, and these cilia are tiny hair-like structures that sweep mucus and trapped pathogens towards body openings. They facilitate their removal through actions like coughing and sneezing. So their main function is like mopping. Okay. And they keep, or they are just like they are giving a flow to the mucus towards the natural openings. Okay. Now you can see here that we have the pathogen as a red, and then we have the mucociliary clearance on this direction on the right side. So here the mucus is secreted by the underlying tissues or glands, and if the pathogen comes here, it is through the ciliary movement, it is washed away, and slowly it goes outside the body. Then we have the tears which wash pathogens from the eyes. So because our eyes are exposed to the environment, and they get millions of pathogens or particles or dust, but this that does not hamper our vision, because our eyes are always moist with tears, and these tears, they wash the pathogens from the eyes. Then urine flushes pathogens out of the urinary tract, and skin cells, they constantly shedding, and the mucosal cells, they remove the attached microbes. So these are some of the very important strategies which our body starts as soon as, or it keeps doing, when any kind of pathogen or microbes, they come in contact. Okay. So ciliary and mucous movement, then tears, then urine and shedding. Right. Now let's see another innate response or another innate strategy, which is the phagocytosis. Now, phagocytosis is the cellular uptake or eating of particulate material, for example, bacteria. And usually phagocytosis happens to the particles which are usually greater than zero point 0.5 micron in size. Okay. Now, this is the next line of defense against pathogens that have penetrated the epithelial cell barrier. So epithelial cell barriers, they are our first line of defense. But if it is breached either by injury or by some other means, and the bacteria or the pathogens, they are successful in invading, then the phagocytosis comes into the picture. Then we have the macrophages, we have neutrophils, we have dendritic cells in tissues, and monocytes in the bloods, which are the main phagocytic cells. So these four cells are the main phagocytic cells: macrophages, neutrophils, dendritic cells in tissues, and monocytes in blood. Now, during phagocytosis, what happens? Here you can see this is the bacteria, and it becomes attached to the membrane evaginations like pseudopodia, and then it is ingested, and once it is ingested, then it forms the phagosome. So this is the phagosome. Now this phagosome is fused with the lysosome, and you know that lysosome contains a number of proteolytic enzymes. And then the bacterium is killed and digested by the lysosomal enzymes, and then the digested products are released from the cell. So this is a very general mechanism of phagocytosis. Okay. Various PRRs or the pattern recognition receptors on phagocytes, they recognize microbes and activate the production of a variety of molecules that contribute in eliminating the infection. So our PRRs are very important, which we are going to discuss today, and these PRRs are present on the phagocytes. Okay, these are basically the receptors which recognize the pathogenic components and they bind with them, and then Once the binding happens, then the production of a variety of molecules are the production of a variety of molecules kicks in, which contribute in the elimination of the infection. Okay, now the related process is the opsonization. So opsonization is the coating of opsonins on the pathogen. So we have the pathogens, and then we have certain molecules which we call them opsonins, which coat the pathogens. So coat means they surround and attach themselves on the outer side of the pathogen. So it indirectly activates phagocytosis. So here you can see this is a pathogen. Here this is an IgG, immunoglobulin G, and this green color particles, these are the complement proteins, and they are again, and these, some thread like are other opsonins, and they have attached on the surface, right? And then there is an addition and ingestion, because we have the receptors here, and once the addition and ingestion happens, then there is a phagosome formation, and ultimately phagolysosome formation, where the lysosome fuses with this particular phagosome, and then it degrades the pathogen. Okay, so this is how the opsonins work. So these opsonins are soluble pattern recognition proteins. These are soluble. These are not membrane bound, but these are free soluble PRRs, which we call them as a pattern recognition proteins. And these bind to conserved repeating components on the surfaces of microbes, such as carbohydrates, lipopolysaccharides, and viral proteins. Got it? So now you know what opsonins are. So basically they are soluble PRRs. And what are PRRs? PRRs are the pattern recognition proteins. So pattern recognition proteins are the receptors or the proteins which, here in case of opsonins, they are freely available, and as soon as they see any pathogen or they interact with the pathogen, they bind with some specific carbohydrates, lipopolysaccharides, or viral proteins, and they coat themselves. So once the pathogen is coated, then it is uh, it is intended for the phagocytosis. So the phagocytosis takes place. So it helps in the phagocytosis. So there are a variety of soluble proteins which function as opsonins. For example, we have collectins, we have SPA, we have SPD, we have MBL, we have L-phycolin, then we have C1q, which are the complement components. Then we have other complement components as well. Then we have IgA, IgG, and then we have the CRP. Okay, so these are some of the examples of the opsonins which are present, and some of these opsonins you must have, you remember that we have just seen in the chemical barriers. They were there in the list in the of the chemical barriers. Now, if we see the opsonin structure, so mannose binding lectin, L-phycolin, and C1q, which is a complement component, they have the similar polymeric structure with collagen-like shafts. So here you can see this is the collagen-like shaft. So here it is the collagen-like shaft. Here it is the collagen-like shaft, and this is the MBL. This is the H-phycolin, and this is the C1 bound to LPS. And but their recognition regions with different binding specificities. Now, what you have to keep in mind is that although their polymeric structure is similar, but they can bind to different their own targets. For example, here you can see the mannose binding lectin. It is binding to the carbohydrate. Okay, the C1 bound to LPS, it is binding to the LPS already bound, and H-phycolin, it is again binding to different carbohydrates. So their targets are different, but their structure is almost similar. They all are bound by the CD91 opsonin receptor. Now, what is CD91 opsonin receptor? It is expressed in multiple cells and key role in intracellular signaling and endocytosis. So once these opsonins they bind the pathogen, okay, then the bound entity they bind with the CD91 opsonin receptors which are present on a number of cells and which facilitates the endocytosis. So their role is very simple. There is a pathogen. Pathogen have certain specific proteins or carbohydrates on the surface. Opsonins are there in our body. These opsonins recognize those specific carbohydrates or proteins on the pathogen. They bind there and they coat the surface of the pathogen, and they on the other side they bind to CD91, which is the opsonin receptors which are present on so many cells, for example phagocytes. And then once they are binding or they are bound to the CD91 opsonin, then the endocytosis takes place, and with the pathogen the phagocytosis process kicks in. Okay, so they help in phagocytosis. Now, if we see the mannose binding lactin and H5 they have N-terminal cysteine-rich region. Okay, so here you can see this is the N-terminal cysteine-rich region here in both the cases, and it is followed by a collagen-like region. So this is a collagen-like region. So this region is a collagen-like region, and this region is a cysteine-rich region. The same thing here in H5 as well. We have the cysteine-rich region, and then we have the collagen-like region. And then we have a recognition domain. Now this is the recognition domain. This is the recognition domain which recognizes the specific targets. So here it is, we have the recognition domain. So here you can see the structure is almost similar. Now both have bound MBL associated serine proteases. We call them as MASPs. So here you can see we have this MASP2, or here also we have this MASP2. So there you can see here the color which is light blue, this is MASP2, and here also we have the MASP2, and MASP2 is MBL associated serine protease. They become active after the recognition domain bind to specific carbohydrate residues on the pathogen. So MSP is present there, but they are not activated unless this recognition domain it binds to the specific carbohydrate residue. So here you can see the carbohydrate. Here is the carbohydrate which is present on the pathogen surface. This is the pathogen and this is the pathogen surface. Okay, I write just P just to recognize that this is the pathogenic surface. And here you can see the carbohydrates are there and they are binding to the recognition domain, and once this binding happens then these MASPs are activated. Then activate the complement pathway, and this complement pathway is a tightly regulated cascade. Okay, we will see in the next class what the complement pathway is, but here you understand that the complement pathway is a tightly regulated cascade which is activated by these opsonins, with and in through this pathway we can detect and tag pathogens for destruction. That is one thing. Then we can recruit and activate immune cells. That is another thing. Then directly lysing the pathogen, another action which we can accomplish through this complement pathway. And then enhancing antibody mediated immunity, again a different function. So complements can do a number of functions, and their one goal is to eliminate the pathogen, okay, through whatever route they are taking, but the ultimate result is that the pathogen is eliminated. Okay, now C1 is what? It is the first component of complement. First component of complement system. It has a structure similar to MBL and H5 So we just saw the structure of MBL and H5 Now C1q can bind to bacterial lipopolysaccharide or antibodies bound to antigens. So if the antigens are there and antibodies have already bound, then CQ1 can bind to these antibodies, or alternatively it can also bind to bacterial lipopolysaccharide. Now here you can see this is C1q. Okay, here it is bound to LPS and here it is bound to IgG. So you can see here this is the LPS and here it is the IgG. and in both the cases C1 has bound. Now after binding, the associated C1R and C1S subunit proteases. So basically this C1 has two subunits, one is C1R and C1S, and these are the subunit proteases which are very similar to MASPs. Okay, so here you can see this is C1R, C1S. So here we have C1R and C1S, and in the previous slide instead of C1 we have the MASPs. So they become activated and initiate complement activation. So again the four functions we described or we just discussed in the last slide, so they become activated and start complement activation. Now we also have the SPA and SPD. Now after binding to microbes, these both SPA and SPD, they are recognized by CD91 opsonin receptor because they are also opsonins. So they are also recognized by the CD91 opsonin receptor, and they promote phagocytosis by alveolar and other macrophage populations. Okay. They clear the fungal respiratory pathogen Pneumocystis carinii, a major cause of pneumonia in individuals with AIDS. So they SPA and SPD, they primarily act in the respiratory system. Okay. Then we have the C-reactive protein or CRP. It recognizes phosphorylcholine. It recognizes phosphorylcholine and carbohydrates on pathogens, and then bounded by Fc receptors for Ig found on most phagocytes, which are important for opsonizing activity of IgA and some IgG subclass. So that's how this CRP works. After binding specifically to antigens on microbe surface, the Fc regions of these antibodies can be recognized by specific FCRs, and it triggers the phagocytosis. So ultimately here also CRP is facilitating the phagocytosis, and SPA and SPD is also facilitating the phagocytosis, and previously discussed opsonins are also facilitating the phagocytosis. Among the most effective opsonins, okay, are several components of the complement system. So complement system, again, this term you are hearing it again and again, but we will discuss in the next class. So complement system is a very beautiful system inside our body, which is composed of a number of proteins, which we call as a complements, and these complements, they do a variety of functions. Okay, so there are so many. So opsonins, one of the best functions of this complement system is that their components, they act as an opsonin, and that is the most effective opsonins. They are one of the most effective opsonins in the body. Now we saw opsonins, we saw the general mechanism of phagocytosis. Now how this phagocytosis happens and what is the killing mechanism, we will see here. So phagocytes, they employ highly toxic reactive oxygen species, which we call as ROS, and reactive nitrogen species, which is RNS. Now these ROS and RNS, they damage intracellular components. How? You can see here we have so many reactive oxygen species. For example, we have superoxide anion, we have hydroxyl radicals, we have hydrogen peroxide, and we have hypochlorous acid. In case of reactive nitrogen species, we have nitric oxide, we have nitrogen dioxide, and we have peroxynitrite. Okay, so here you can see the oxygen using the NADPH phagosome oxidase. Now this is important, NADPH phagosome oxidase. This is very unique to phagocytes. This is unique to phagocytes. So this is using this oxygen, and this oxygen is coming as a respiratory burst, right? So this oxygen is provided by a metabolic process which is known as respiratory burst, where the oxygen uptake by the cell increases several fold. So this is a kind of a metabolic reaction. We call it as a respiratory burst, and suddenly there is a gush of oxygen, and when there is a gush of oxygen, then this NADPH phagosome oxidase, which is unique to phagocytes, it forms superoxide anion. right? And this superoxide anion, it can, in the presence of superoxide dismutase, it forms the hydrogen peroxide. And this hydrogen peroxide, it produces the hydroxyl radicals as well as with the chloride ions, and using myeloperoxidase is also produces the hypochlorous acid, and these are all toxic. Now here if you see, we have L-arginine and we have inducible nitric oxide synthase, which is INOS, right? Its expression is activated by microbial components binding to various PRRs. Remember, I-ionase is activated by microbial components binding to various PRRs. So once this inducible nitric oxide synthase, it gets activated, it converts L-arginine to nitric oxide with the expulsion of L-citrulline. And this nitric oxide with it forms the nitrogen dioxide, and this nitric oxide also with superoxide anion, it produces peroxynitrite, another uh, reactive nitrogen species. So you can see there are so many reactive nitrogen species and reactive oxygen species which are generated during this process, and they are highly toxic to the pathogen, killing the pathogen inside the cell. So ROS and RNS also can be released to kill extracellular pathogens from activated neutrophils and macrophages. Now this is important because now till now, what we saw is the intracellular metabolic pathways here. But once these ROS and RNAs are generated, they can not only kill the intracellular pathogen, but also they can be released and they can kill the extracellular pathogen as well. Okay, this is very important point here. And then we have the enzyme cyclooxygenase or COX-2. It converts lipid intermediate arachidonic acid to prostaglandins, which are potent pro-inflammatory mediators. So once the prostaglandins are formed, then the inflammasomes or the inflammation reaction, it gets activated, and then the inflammation also helps in eliminating the infection. Okay, now macrophages are the main scavenger cells to clear the cellular debris also, right? So we have a lot of cellular debris during during the growth or during the metabolism. So macrophages are the main cells to clear the cellular debris also. Now it recognizes the components of the dead or dying cells and the damaged tissue, and these we call it as damage associated molecular patterns. We call it as a DAMs. Okay, what are DAMs? DAMs are the damaged associated molecular patterns, and these are the components of the dead or dying cells and the damaged tissues. So once these DAMs are recognized by PRRs, then it leads to their clearance, and through the process of phagocytosis, these macrophages, which are the main scavenger cells, they clear the tissue from any debris. But the important point here is that they should, the components of the cellular debris, which we call them as the DAMs, they should bind to the PRRs of the macrophages in order to activate the macrophages. Okay. Now, apoptotic cells, they attract phagocytes by releasing the lipid mediator lysophosphatidic acid. Lysophosphatidic acid. So these are released by the apoptotic cells. Apoptotic cells are those cells which have undergone or which are undergoing the programmed cell death. Okay, and once this process of apoptosis it kicks in, then there is no U-turn, and then the cells release this lysophosphatidic acid, and it attracts these phagocytes, and these phagocytes, they start eating the components of the apoptotic cells. So basically this phosphatidic acid, it functions as a chemoattractant of phagocytes. Okay. So if we see the dams of the apoptotic cells, so what are dams? Okay, just dams are the damage associated molecular patterns, which are the components of the dead or dying cells. So if we see the different dams which our macrophages they recognize through PRRs, they are phosphatidylserine, they are this, then another is the lysophosphatidyl then we have annexin 1 protein, then we have altered carbohydrates, and in case of phagocytic receptors we have phosphatidylserine receptor and we have scavenger receptor SRA1. So these receptors they recognize these dams, and once the interaction is complete then the cell gets activated and the clearance of the apoptotic components or the damaged tissues are done. Okay. Some dams are recognized by opsonins activating phagocytosis. So suppose few dams they are not able to bind to the macrophages. Then the opsonins, because they also have the they also have the affinity for these dams, so they bind these dams and then they initiate the phagocytosis. Now if you see, we have the young and healthy RBCs. Our young and healthy RBCs avoid phagocytosis by not expressing, okay, not expressing eat me signals. So eat me signals are the signals which are expressed by the dying cells or the damaged tissues, which says, oh come eat me. But these cells, the young and healthy RBCs, they avoid phagocytosis by not expressing the eat me signals, for example phosphatidylserine. And in the last slide you remember, in the list of the dams phosphatidylserine was there, and it also expresses a don't eat me signal, which is the protein CD47. So CD47 is a don't eat me signal, and the eat me signals are phosphatidylserine. So these are absent and CD47 is present, and that's how our RBCs they avoid phagocytosis, right? But once CD47 is expressed, then it is recognized by SIRP alpha receptor. So here you can see this is SIRP alpha receptors on the macrophages, and here you can see this is the high CD47 density or clustering. So these are CD47, and this signal says don't eat me. So it is recognized by SIRP alpha, which transmit signals that inhibit phagocytosis. So it will not going to phagocytose these RBCs. Now tumors use this elevated CD47 expression. This is the same CD47 expression to evade tumor surveillance and phagocytic elimination. So this is the strategy which is adopted by tumor cells to evade phagocytosis, that they express high level of CD47, and this high level of CD47 when it interacts with the SIRP alpha receptor on the macrophages, then macrophages think that no, I don't have to eat this while they are the tumor cells. Okay. Now, increased expression of CD47 on human cancer is correlated with tumor progression also. So this CD47 is also one of the markers. In the patients, if the CD47 is increased, that also correlates that the tumor is progressing. Now, some bacteria like Listeria, they skip phagocytosis by replicating in cytosol. So they come in cytosol, they replicate easily, they don't go inside the endocytosis. So therefore they are basically avoided. Now they are eliminated by another process which is known as autophagy. This is the intracellular phagocytosis. What is autophagy? Autophagy is the intracellular phagocytosis. So membrane derived from the endoplasmic reticulum envelopes the bacteria forming an autophagosome. So here you can see we have the membrane which is derived from the ER envelopes. Okay, and then we have the bacteria or pathogen, and this membrane it grows and it completely envelops this pathogen and it forms this autophagosome. Now this autophagosome it fuses with the lysosome and it forms the phagolysosome, and then again the same process, the enzymes they start degrading this pathogen and ultimately you can see the lysis of the bacteria or the pathogen. Now this innate effector function is activated by NLR, which is the node like receptors which we will be seeing, and it's a kind of PRR as node 1 and node 2. Okay, we will study it after a short while. Now, this slide talks about the effectors of the innate immune responses. So, whenever there is a pathogen, now you can see here we have a pathogen. Then we have the phagocytes, right? Then we have the antimicrobial peptides. We have the complement proteins, CRPs, SPA, SPD. These are the opsonins, okay? And then they opsonize, and then they, help killing the bacteria. They antimicrobial and peptides, they are also killing the bacteria, and through phagocytosis also the bacteria is killed. So basically we have these effector molecules and the effectors which are which play important role in the innate immune responses to infection. Now, in case of innate initiation of adaptive responses, we have the DCs or the dendritic cell PRRs. They recognize PAMs, they activate phagocytosis and signaling pathways. And then these DCs or dendritic cells, they migrate to lymph nodes with the antigenic peptide and present it to the T cells, which recognize the antigen MHC complex, and ultimately the T cells get activated and our adaptive response also kicks in. So that's how this is called as the cooperation between these two branches, the innate and the adaptive branch. Now, let us focus on PRR PAM. PRR is the pattern recognition receptor, and PAM, in the first video lecture also, we touched upon this. PAM is the pathogen associated molecular pattern. So, pathogen associated molecular patterns are present on the pathogens, while PRRs are present on our own cells. And this PRR PAM based recognition is the key, and it is a very effective and very important aspect of the innate immune system. So now see carefully. Innate cells, they express a variety of receptors, and these are called as pattern recognition receptors. We call it as a PRRs, which are the receptors present on the innate cells, right, which recognize pathogen associated molecular patterns, which call them as PAMs, and damage associated molecular patterns or DAMs. We just saw the DAMs. Okay, now PAMs are specific conserved molecular components on the surface of the microbes. They are present on the microbes, but they are absent on our own cells, so they are easily recognized. More general term for PAMs is microbe associated molecular pattern or MAMS. So if we are using the term PAM or we are using the term MAMS, both are same. But PAM is a bit specific because PAM is associated with the pathogen, while MAM is associated with the microbe. And why? Because some microbes are non-pathogen too. Not necessarily that all the microbes are pathogenic. Some microbes are non-pathogens too. So that's why we use a broader term, which is MAMS, or the microbe associated molecular pattern. So here you can see this is the PRR, and this PRR is a receptor which is present on, let us say, this is a macrophage, and here you can see we have the DAM and here we have the PAM. In both the cases they can bind to PRR, and once this PAM and PRR bind, we call it as a PRR PAM interaction. Once this PRR or DAM it binds to the PRR, then it activates the cell, and then the elimination of the microbe or the pathogen it starts, right? Some PRR triggers other responses. Not only necessarily the phagocytosis is the only way, but there are certain other responses as well. Now, if we see the PRR PAM based recognition, many PRRs are expressed on or inside cells. So, there are certain PRRs which are present on the membranes. For example, we have TLRs, we have CLRs, they are the membrane bound PAMs. But there are certain, PRRs which are present inside or the intracellular also. So, here you can see NLR, RLR, C gas, these are, or some TLRs, they are also present inside the cell so that they can recognize the components of the microbes, for example the viral RNA or DNA, right? So PRRs are commonly expressed by all types of myeloid WBCs, all the WBCs like monocytes, macrophages, neutrophils, eosinophils, mast cells, basophils, and dendritic cells. They all express these PRRs. Then we have the subsets of lymphocytes like B cells, T cells, and NK cells. They also express the PRRs. Then some cells which are commonly exposed to infection agents, for example, skin, mucosal and glandular epithelial cells, and vascular endothelial cells or fibroblasts and stromal cells, they also express the PRRs. So you can see we have a wide variety of cells which are expressing the PRRs, and their only aim is to catch the pathogen which is expressing PAMP or MAMP. Okay, and in our own bodies or some healing is required, then DAMs. So once DAMs, MAMs or PAMs they recognize or they detect, they immediately bind and they activate the respective cells, and most of the times it is the phagocytosis, but there are other ways also through which the pathogens are eliminated. Okay, so PRR PAMP recognition is the major key or the major mechanism in innate immunity. Okay, now if we see the PRR families in our body, we have the toll like receptors which we call it the TLRs. The key member are TLR1 to TLR10 in case of humans, and TLR11 and TLR13 also present in case of mouse. Their location is either cell surface or endosome. So they are extracellular as well as the intracellular. Main ligands are lipoproteins, LPS, flagellin, nucleic acids, and the pathogens they sense are bacteria, virus, fungi, and parasites. Almost all classes of the pathogens. Then another family is the C-type lectin receptors or CLRs. Examples are dactin 1, dactin 2, Minkel, and DC sign. They are present on the cell surface, and their main ligands are carbohydrates, mannans, beta glucans, and they sense fungi, some bacteria, and viruses. Then we have the node-like receptors as a third family. We call them as NLRs, and the key members are node 1, node 2, NLRP3, NLRC4, etc. And their localization is cytoplasm, and their main ligands recognized are peptidoglycans, PAMs, and DAMs. And the pathogens they sense are bacteria or viruses. Then another family is RIG1 like receptors or RLRs. And the examples are RIG1, MDF5, LGP2, and they are present in cytoplasm and they detect the viral RNA. So viruses are their major targets. Then we have the ALRs, which is the AIM2 like receptors, and examples are AIM2 and IFI16, and their main location is the cytoplasm and the nucleus, and they detect the cytosolic DNA. So viruses and bacteria are their main target pathogens. And then we have the C gas sting pathway. This is also kind of PRR family, we say them, and the major components are C gas and sting. Then again, their location is cytoplasm, and their main ligands are cytosolic DNA, and they also recognize the viruses and bacteria. So you see overall we have as per our current understanding, we have the TLRs, we have CLRs, we have NLRs, we have RLRs, we have ALRs, and we have C-gas sting. Total 6 types of PRRs in a family form. Okay. Now we will see, each family slightly in details and how they recognize that. Okay. Starting with the toll-like receptors or TLRs. So this is a family of type transmembrane proteins, right? Type transmembrane proteins. It's a kind of a, transmembrane proteins. Common structural elements they have is the leucine-rich repeats. Every TLR has LRRs in their extracellular region. Cytoplasmic toll or IL-1 receptor domain mediating downstream signaling, and then they dimerize upon binding the PAMP or DAMP. This is the characteristic or their feature. So if you can see here, this is the leucine rich repeat, which is the LRR, and this is the exterior domain, okay, in the extracellular region. And then we have the TIR domain, which is the tall IL-1 R receptor. This is TIR domain. It is in the cytoplasmic tail, as a cytoplasmic tail, and it mediates downstream signaling. And the feature of this toll-like receptors are they dimerize upon binding to PAMs or DAMs. Okay, so how they dimerization happens, we will see. Humans have 10 functional TLRs, and these are TLR1 to TLR10, while mice have 12 functional TLRs, TLR1 to 9, then 11 to 13. TLR10 is non-functional in case of mice. Now TLRs 1 to 10 are conserved in human and mice. Okay. Now cell membrane TLRs are TLR1, TLR2, TLR4, 5, 6 and 10s. They detect PAMs, they detect DAMs, while intracellular TLRs are TLR3, 7, 8 and 9 on endosomes or lysosomes, and they sense foreign nucleic acids. Okay. TLR4 is very unique. It is found both in the plasma membrane and in endosome. So this TLR4 is the TLR which has been found on the plasma membrane as well as on the endosomes. Now these TLRs, they trigger signaling cascade leading to the activation of transcription factors like NF-kappa B, AP1, CREB and IRFs. Okay, so their ultimate function is to activate the transcription factors NF kappa B, AP1, CREB and IRF. The result is the production of pro-inflammatory cytokines. Okay, once what do? These transcription factors, they produce the pro-inflammatory cytokines, they produce chemokines and type 1 interferons. You remember in our last discussion in the cytokines, all these things came, and these coordinate the immune response to eliminate pathogens and initiate tissue repair. So that's how they work. TRR signaling also bridges innate and adaptive immunity by activating antigen presenting cells and promoting the maturation and activation of T and B lymphocytes. So it also bridges innate and adaptive immunity. Again, it is a sign of cooperation, example of cooperation. Abnormal TLR signaling has been implicated in various diseases, for example, sepsis, autoimmune disorders, allergies, cancer, and chronic inflammatory conditions. If there is some abnormal TLR signaling, it may lead to diseases as well. All right. So here, this is a snapshot summary of toll-like receptors. Now, if you see the toll-like receptors here, this is TLR2, TLR1, okay. This is a heterodimer, this is heterodimer TLR2, TLR6. This is homodimer of TLR5, both of the molecules are TLR5, and this is TLR4. So different TLRs may be differentially activate distinct transcription factors, leading to variation in which genes are turned on to help protect us against invading pathogens. Now here you can see TLR4. Then we have the in the endosome we have TLR3, 7, 8, and 9. And now there these TLR2, TLR1, 6, 5, and 4, they recognize membrane components. Okay, their main function is to recognize membrane components. So you can see here TLR2, TLR1, it recognizes triacyl lipopeptides. TLR2, TLR6 recognizes diacyl lipopeptides. TLR5 recognizes flagellin, and TLR4 recognizes LPS. So they recognize the membrane components. Now, if you see the endosomal TLRs, they localize to endosome and recognize nucleic acids. And this nucleic acid can be from host as well as the foreign nucleic acid. Okay. And TLR4 is present at the membrane but endocytosed upon activation. So once it is activated, it dimerizes. So you can see this is the homodimer of TLR4, and once it dimerizes, then it is endocytosed upon activation. Clear? Now, there is a ligand binding. So these ligands, they bind to the TLR. Then TLR dimerizes. So all the TLRs, they become dimerized. Okay. Then there is a TIRAP and MyD88 binding at the TIR domain. So here you can see the TIR domain, and then there is a recruitment of these two proteins with TRAM or TRIF. Okay, either direct or TRAM or TRIF. So here you can see this is the direct TIRAP MyD88, but here it is with TRAM or TRIF. Okay. In endosome, TLR4, this is endosome, this is TLR4 in the endosome. It switches from MyD88 to TRIF. So you can see here it was initially when this TLR4, it was here, right? It had the MyD88. This you can see the MyD88. It was here. But once it is endocytosed, then it switches from MyD88 to TRIF. This. Okay. So this is the difference when it is on the surface and when it is endocytosed. So now they make MyDDsome by complexing with IRAK4 and IRAK12. So they make the whole MyDDsome. And then this MyDDsome, it activates the TRAF6. Okay, it activates the TRAF6. Now, this TRAF6 also activates TAC1 complex. TRF6 also activates this TAC1 complex, and you can see that this TAC1 complex, it activates different type of ultimately pathways which we have discussed in the cytokines, and they form or activate the transcription factors here, and these transcription factors, they ultimately give rise to the pro-inflammatory cytokines or type 1 IFNs. So that's how the toll-like receptors, they do the signal pathway, and then they activate the transcription factors, respective transcription factors, and these transcription factors, they generate the pro-inflammatory cytokines or different other kind of chemokines or cytokines. Now, the second family is C-type lectin receptors or CLRs, right? Now we have the dactin 1, which we call it as a CLEC7A, and its ligand is beta glucan, which is present in the fungal cell walls. The main function is the fungal recognition, phagocytosis, cytokine production, and T cell polarization. The main signaling motif or pathway is ITAM like motif and SYK card 9 NF kappa beta. So these all things we have already discussed in the cytokine class. Then we have the dactin 2. Its ligand is alpha mannans, which is again a fungal cell walls, and their main function is the fungal recognition, cytokine production, and Th17 response. And signaling motif of pathway is the associates with FCR gamma, which is ITKM and SYK. Then we have the Minkle, which CLEC4E. Its ligand is SCP-130, which is produced by the dead cells, and then some mycobacterial glycolipids. It sends cell death, inflammation, antifungal, and antibacterial responses. It associates with FCR gamma, which is again ITAM, SYK-SIC, and PLC gamma 2 and NFAT, right? And then we have the CLEC9A, which ligand is actin from necrotic cells. It sends cell death, cross presentation to CD8 positive T cells, and immune regulation. And their signaling motif is Hemi, ITAM, SIC, and SHP1. So they have at least one C-type lectin-like domain to recognize a diverse ligands, including carbohydrates, proteins, lipids, and altered self proteins. Okay, so actually, you must be feeling a bit bored here because we are discussing the different CLRs. But what I want to emphasize here is that the different components of the families, they have their specific or some broader roles. Now here, if you can see, they are dactin 1. It is for beta glucans for fungal cell wall. But here, if you see dactin 2, it is also the fungal cell wall, but here the main target is alpha mannans, right? So there is a difference that what is recognizing what. Now, we have the whole array of these proteins, and these proteins are capable to recognize the different components on a same cell. And once it happens, then the immune response is very effective. That is the whole purpose of discussing these minute details here. So if you see in nutshell, these C-type lectin receptors, so CLRs, they are the diverse membrane receptors, and they are present on monocytes, macrophages, DCs, neutrophils, and B and T cells. Now again, if you see here, we have CLEC12A, then we have DC sign, then we have mannose receptor, then we have Langerin or CD207, then we have CLEC5A or MDL1, and we have CLEC4D. And here you can see different ligands, their main functions, and signaling or motif pathway. So you can slightly pause this slide, and then you can take a note out of this. Now if we see the C-type lectin receptors signaling pathway, then you can see here we have the fungal cell. Okay, now this is the fungal cell here, and on the fungal cell we have the glucans. This is the fungal glucan. Now this fungal glucan, it binds to dactin 1 dimer. So you can see here, this is the dactin 1 dimer here. Now phosphorylation of the tyrosine in the half ITAM. So you can see here, this is the half ITAM, which is the immunoreceptor tyrosine based activation motif. It initiates a signaling pathway that activate transcription factors NFAT, here it is NFAT, then NF kappa B, here it is NF kappa B, we have IRF5, and then we have AP1. These are the different transcription factors which we discussed in details in our cytokine class, and you can just refer it again there. And then their main job is to activate the transcription of interferons, cytokines, chemokines, and antimicrobials. So unlike TLRs, which do not promote phagocytosis, some CLRs function as phagocytic receptors. So that is slight difference from the TLRs if we compare the TLRs and CLRs. Then we have the third family, which is the retinoic acid inducible gene 1 like receptors, or we call them as RLRs. They are intracellular soluble PRRs of many cells. Now here important is, number one, they are intracellular. Okay, so they are not membrane bound. Then external membrane bound. Then they are soluble. Okay, so soluble means they are freely available inside many cells. In innate antiviral immunity. Okay, in addition to TLR7 and TLR9 based viral nucleic acid recognition, Most cells recognize viral nucleic acids through these RLRs. So these RLRs are very, very important for the viral nucleic acid recognition. The three RLRs, we have the RIG1, MDA5 and LGP2. They are CARD containing RNA helicases. So we call them as a card containing RNA helicases. And these helicases recognize viral RNAs. And most of the times, and specifically, they recognize influenza, measles, and West Nile. These are the examples of the RNA viruses. Right now, these viral RNA binding leads to RIG1 conformational change. Okay, here it is the RIG1, and then there is a change which further binds via card-card interaction to its adapter molecules MAVS. So here you can see it binds to MAVS. This is MAVS, which is the mitochondrial antiviral signaling protein in mitochondrial membranes. Now these MAVS, they aggregates and recruit additional proteins including the adapter TRADD, TRAF3 and RIP1. So you can see here it is aggregating TRA, DD, RIP1 and TRAF3. Alright. Now this activate Nemo IKKs that lead to the activation of IKK complexes which activate IRFs 3 and 7 and NF kappa B pathway to express IFN alpha and beta, some other antimicrobials, chemokines, and pro-inflammatory cytokines. So here you can see here the end result is the production of interferon alpha, interferon beta, and some antimicrobials and chemokines. Okay. Now, another family or the fourth family is NLRs or the node-like receptors. These are again intracellular PRRs, okay, and they have the three domains. We have the central nucleotide binding domain, okay, or NACHT domain. This is called as NACHT domain, which is the abbreviation of the four kind of NLR members. We have four NLR members. We have NAIP, CIITA, HTE, and TP1. So we, abbreviated all these fours and we made a name which is NACHT, NEST domain or NACT domain, okay, for nucleic acid binding and oligomerization of NLRs. So this is the central domain, okay. Then LRRs at the C terminal. So you can see here, this is the LRR and this is the C terminal. Let me write it, C terminal, to identify ligands, and N-terminal effective domain. So here this is the effector domain and I can write it, this is the N-terminal domain. So basically this N-terminal effector domain, it's a protein interaction domain and caspase activation and recruitment domains or the PYD domain, right? So card or pyrin domain or PYD domain. So PAMs, PAMs. PAMs are cytoplasmic pathogens, components and of infection and injury. We just, discussed. Then we have the five subfamilies, and these five subfamilies are NLRC subfamily, which contains cards. We have NLRP subfamily, which contain PYD. If you recognize here NLR remains the same, but last letter it recognizes what it has. So NLRC, it contains card, C card. Then NLRP P, it contains PYDs. Then we have NLRB subfamily, which contains baculovirus inhibitor of apoptosis protein repeats. Then we have NLRA subfamily, which contains acidic activation domains, and we have NLRX subfamily, which contains the other NLR effector domains. So we don't recognize any, so we have written X, right? So these are the five subfamilies, okay? So ligand binding changes the conformation of NLRs from self inhibition to activation. So here you can see they are, you know, their conformation is like this is in a inhibition mode, and once they bind with the PAMP, so PAMP is binding to LRR, then immediately it gets activated, and once it gets activated, then you can see here dimerization happens. So U-shaped configuration, it inhibits its multimerization, right? So it remain as a single entity because this is the U-shaped configuration. But PAMP binding changes the conformation and then triggers the oligomerization to activate the NLR. Now it is important that to activate we need this oligomerization. If it is not oligomerized or dimerized, then this will not get activated. Okay, the exposed and terminal effector domain, so you know this is the effector domain, the pink one, right? The exposed and terminal effector domain through homotypic interactions, downstream adapter molecules and signaling protein initiate the signal transduction. So ultimately they get activated and the downstream signal transduction process takes place. So NOD1 and NOD2 proteins are the most studied protein of the NOD-like receptors or NLRs. That's why we call this as a NLR family or NOD-like receptors family. NOD1 recognizes diamino pymalic acid of the gram-negative bacterial cell wall, and NOD2 can recognize muramyl dipeptide in all bacterial cell walls and a complete viral single stranded RNA. Now this is important, not the piece but a complete viral single stranded RNA. So you can see, what is the range of the recognition these NLRs can do. NLR1, or sorry, this NOD1, it can recognize this particular component in gram negative bacterial cell wall, while NOD2 can recognize muramyl dipeptide in all bacterial cell walls and a complete viral single stranded RNA. So NOD1 and NOD2 eliminate cytosolic bacteria by autophagy as well, right? So they are not only binding to the viral single stranded RNA but also to the bacterial components, and therefore the bacteria which want to hide inside the cytosol and they don't get phagocytosed, they are eliminated using NOD1 and NOD2. So they are very, very effective for intracellular pathogens. Okay. Some NLRs, they are the part of inflammasomes. So we will, see inflammasomes in next class, but some NLRs, they are the part of the inflammasomes which actually kicks in the inflammation reaction and are responsible for the inflammation. A very effective mechanism in innate immunity. Then we have the ALRs or AIM2 like receptors. These are the new type of PRRs which can recognize intracellular foreign DNA. Okay, ALRs can recognize intracellular foreign DNA. So we have the C terminus DNA binding domain, right? So this is the C terminus and this is the DNA binding domain, which is HIN, which is hematopoietic expression, interferon inducibility, nuclear localization 200. And the N terminus is the PYD domain, pyrin domain, yeah, which is the N terminal. This is N and this is the C terminal. Now sometimes it referred to as a PYHIN family. We also call it as a PYHIN or PyHIN family. The HIN 200 domain recognizes double-stranded DNA and binds to it. So the main function of this HIN 200 domain is to recognize double-stranded DNA and then it binds to it. Okay. The N-terminal PYD binds to the PYD of apoptosis associated SPECT-like protein containing card, which is like for example ASC, and they promote the formation of inflammasomes and the maturation and release of IL-1 beta and IL-18. So that's how once they bind, then this PYD domain, it promotes the formation of inflammasomes and the release of IL-1 beta and IL-18. It can also regulate apoptosis in some instances, right? Now the last is the C gas and sting. They are recently discovered cytosolic PRRs. Again, these are cytosolic, not membrane bound. And C gas is the cyclic GMP AMP synthase. They recognize cytosolic foreign DNA. So they also recognize cytosolic foreign DNA. After C gas binds double stranded DNA, it is activated to synthesize the dinucleotide cGMP from GTP and ATP. Okay, so once the C gas binds double stranded DNA, it is activated to synthesize the dinucleotide cGMP from GTP and ATP. So here you can see cGMP from GTP and ATP. Here it is GTP, here it is ATP. It's binding with the DNA. Now cGMP binds to the ER associated protein sting. So endoplasmic reticulum, here you can see, and there we have the sting protein. This sting protein is present on the endoplasmic reticulum, and this cGMP, it can bind to this sting, which is the stimulator of interferon genes. Sting, the full form of sting or the abbreviation is stimulator of interferon genes. Sting can also bind the cyclic dinucleotides released from intracellular bacteria. So some intracellular bacteria, they also release cyclic dinucleotides, and this sting can also bind to it. Okay, so here you can see this is the cyclic dinucleotides which are generated by this cytosolic bacteria. So they are also bound here. Binding alters the sting dimer's conformation to recruit and activate TBK1. So here you can see it can recruit TBK1. TBK1 phosphorylates and activates IRF3 and IKK. Okay, here it is IRF3. You can see the phosphorylation, and IKK, you can see the phosphorylation, which activates NF kappa B. Okay, here it is NF kappa B, and leading to the synthesis of type 1 IFNs and cytokines. So their goal is to produce the type 1 IFNs and cytokines. Okay, so this discussion was about the 6 families of the PRRs and how these PRRs are placed, while on one way we see the TLRs which are present on the membranes as well as inside the endosomes, while the later families of PRRs, they were exclusively inside the cells, intracellular, and they were just meant for elimination of the intracellular pathogens. So now, by now you must have seen that the innate immunity is not only takes care of the extracellular pathogens, but also it has so much to eliminate the intracellular hidden pathogens also. Okay, and the last discussion, so the last discussion about the PRRs has proven this thing, that our innate system is very adequate and very elaborate system. It is not a easy system, or it is not a system something our pathogens can easily skip. But wherever they will be inside the body, whether in the tissue or inside the cell, they will be recognized, they will be detected, and they will be eliminated. Okay, now the last part of today's discussion is about the regulated cell death. You know that we have apoptosis as a regulated cell death, right? But there are some other mechanisms also, we will see here shortly. So PAMP PRR signal, it can induce regulated cell death that can control infection, right? Now one is the apoptosis. Apoptosis is by tumor necrosis factor binding to TNF receptor and then it is mediated by natural killer cells and T cytotoxic cells. Okay, so this can control infection, so it can kill the cells, our natural killer cells and TC cells. They can kill the cells using the TNF, and this mechanism is known as apoptosis or the programmed cell death. Second is we have netosis. Okay, what is netosis? It is the activated neutrophils, they expel. Okay, expel means they take out the chromatin filaments. They have the chromatin filaments, so they simply expel their chromatin filaments. We call them as a neutrophil extracellular trap or NET. This NET term comes from this neutrophil extracellular trap, and antimicrobial peptides to entrap and kill pathogens. Now you can see here, these are the neutrophils which are activated by PAM, which are the pathogen associated molecular patterns. Now they are expelling their chromatin filaments as well as the antimicrobial peptide to trap these pathogens. These red colors are the pathogens. They are easily trapped here and then killing them. Okay, but during this process, these neutrophils are also dead. Okay, so basically this is a kind of suicidal attack on the pathogens. Okay, then we have pyroptosis, the third kind of regulated cell death. So pyroptosis is induced by inflammasome, and inflammasome are the protein complexes that activate caspase 1 to generate IL-1 beta activation. Now caspase 1, you remember that it's a part of apoptosis, right? The death of the infected macrophages, it prevents spread of intracellular bacteria, salmonella and listeria that replicate in these cells. So again, the macrophages are sacrificing themselves because the bacteria listeria and salmonella, they are present inside the macrophages, and now in order to spread these pathogens, the macrophages they are dead using this technique which is called as pyroptosis. They induce inflammasome and then activates the caspase 1, and this caspase 1 generate IL-1 beta, and ultimately that leads to the death of the infected macrophages, not the normal macrophages but the infected macrophages. So that means these infectious agents, they trigger the pyroptosis. Okay, the release of mature IL-1 beta and IL-18 is generated by inflammasome. They mediate cleavage from large precursors, and they are very important pro-inflammatory also. So it not only eliminates the cell, but also they induces the inflammation, local inflammation at the site of the injury or at the site of the infection. Okay, so now we have we are at the end of this class, and let us summarize what we learned today. So we discussed the properties of the innate immunity, then we saw the four barriers of the innate immunity: the physical barrier, chemical barrier, biological barrier, and then flushing. Okay, then we saw phagocytosis, its mechanisms, and how reactive oxygen species and reactive nitrogen species, they are generated and they kill the pathogens. Then we also discussed about the opsonization, and with the few examples of opsonins, and how this opsonins, they act upon the pathogens or microbes and they help in their elimination. And then we discussed the 6 families of PRRs, and the different mechanisms and the signal transductions in detail. Apart from that, we also saw the, as a last slide, the various type of cell deaths, okay, which are associated in the immune response. So here we are ending the part 1. Now in the next class we will be starting with the further discussion about the innate immunity, and that will be in the part 2 lecture. By the time, take care of yourself and thank you.